would it be possible for you to just educate the audience on the difference between, because you mentioned this word multiple times, PCR, and sometimes I know we know what it is here, but maybe you could kind of elaborate on, let's say, PCR versus the technology that we're speaking about, this so-called next generation sequencing, and just maybe contrast those two for the audience first, and then I'm sure it causes the big data questions.
Well, thanks for that, Les. That's one of my favorite questions because I've obviously worked in NGS, but also worked in PCR, and I think they're both amazing technologies. But for a long time, and PCR is much older. I mean, the first versions of PCR was invented by Kerry Mullis in the 80s. Famously got the Nobel Prize for it. Um, and PCR is really a harnessing of nature. Going, DNA comes in a double helix, and cells are really, really good at replicating it. You kind of unzip it, you do some magic. Obviously, we won't go into it in detail here, but Mullis's genius was recognizing a way to do it in a test tube, that you could do it. You didn't need all the mechanisms of a cell to do PCR. And so he worked out that you could, with a very simple apparatus, you could magically replicate DNA. And that turns out to be so useful right across the world because in engineering terms, it's amplification. You just give me one of these things and I can multiply it by a billion because it doubles every time. And it's that classic, one piece of rice on the first square, two pieces of rice on the second chessboard square, and you end up like with a whole universe full of molecules by the time you get to the 64th square. And so PCR is just an amazing amplification effect of DNA inside a test tube, and you know the field has worked out just so many different ways to harness that. And I think most recently, most famously, it became famous during COVID because it's an amazing way to do a COVID test. And the COVID test is actually a PCR reaction which only fires up in a blood sample or a spit sample. It only fires up if it finds the very set sequence, like a subsequence of the viral genome of COVID-19. And then it just goes bam, and it makes a billion copies of it. And you can think of each one having a little tiny LED attached to it in the sense that a little bit of light suddenly turns into a massive amount of light. And that's the test. It's just replicating the DNA of the virus or RNA of the virus. And then sufficient that there's enough light that comes out of the sample that it can be seen on a camera, and that's a positive test. NGS, on the other hand, is so I always say PCR is great when you know what you're looking for. If you've got a sequence, I have a DNA sequence, and it might be just 10 or 100 bases long, and I want to find out if it's in a sample. It's a quick and easy and incredibly sensitive and accurate way to do that. NGS is infinitely more complicated, in effect. Just one small step of NGS is a PCR step, for example, plus many, many other things. But it is able to actually determine the sequence. And I like to use this analogy. If it's the difference between reading a novel and using a search engine in a novel, right? NGS is reading the novel. Like, I haven't read it before. I'm going to read the whole thing. I'm going to learn lots. It's going to take me a lot of time. But if I want to know, does the word universe appear in the novel? How many times is it? I use a search engine, right? And that's what PCR is. So they're fundamentally different. They're very complementary. I think in particular in COVID, NGS decoded the COVID sequence, which enabled everyone to build a PCR test, which could then detect it all over the world. And if a PCR test is at least 100, if not 1,000 times less expensive than an NGS equivalent test. But many would dispute that. But I would say let's say anywhere between 100 and 1,000 times cheaper.